reason why we still observe in the scientific literature on such different results. What we got from the interventional study and from the geodata set, as I did in my work, are not presenting the history and the comorbidity of the cohort they are studying. So how do we know that in some cohort ACE2 is even increased? So it's in apparent contrast what I presented today, but also to that beautiful session I was able to attend yesterday. I don't know if some probably of the audience attended also the preclinical session of yesterday. So how do we know that it's not the comorbidity that is upregulating ACE2 in the lungs? Because as we know, physiologically ACE2 has an anti-inflammatory effect. So that could be a compensatory effect in the chronic smokers. But if we want really to dissect the mechanism of smoke on the risk of COP2, then probably the in vitro study with the ISO standard as we did, then it's probably more able to elucidate the real mechanism. Also, another technical problem: how do you smoke how do you let yourself smoke? I see some colleagues, they use bubbling and then they put the media directly into the cells, but that is not the way we want to expose the epithelial cells to cigarette smoke. So we need to have a real-life exposure of cells. That is what we can do with smoking machine of any kind. We just use the one from Borgwald, but there are many others on the market. They allow you to select the correct regimen.
Thank you. So we had a presentation earlier that talked a little bit about sex differences in scientists. So we have a question from the audience as to whether the cells in the study were derived from men or from women.
The issue, because we know that hormones are direct effect, especially on the protease. That is the second step following virus binding to the ACE2 receptor. Then the protease let the virus inside the cells. And we know that the activity of this protease in a direct effect on hormones and testosterone. That's why we had a more prevalence of men compared to women on COVID-19 infection. And also again, in the cohort we selected, some of these data are lacking. So this is another bias that we need to consider when studying geodata set. But the one that we used from men, H292.
Thank you. I have one more question for you that I'm typing into our chat, and I'm going to read it for the audience so they have the question. So, is the H2 data on smokers versus non-smokers the mRNA data? If so, that would be consistent with the earlier presentation where the protein expression can have a negative feedback on the mRNA.
Absolutely, yes. There are data from the transcriptomic analysis on the geodata set, and this is why also we decided to perform our experiment with high content screening following segmentation of the cells. Because, of course, we want to measure just the expression of the protein on the membrane, because we know that this protein is then internalized. So even Western blot, even if we are measuring protein, would not be sufficient, because we would probably get the same amount of protein. So we just want to measure the one on the membrane, or at least to separate with centrifugation membranes. But since we had high content screening apparatus in our lab, we prefer to use that one and to segment the cells by specific dye of membrane with the cell mask from molecular probe.
That makes sense. Thank you so much. We're going to go on to our next presentation, which is number 4 in our agenda for today. And